All right. So in this video, we're going to address some of the topics we didn't quite get to on Wednesday. We are also going to address a paper that I will post on Canvas as soon as this is over. And on Friday, we're actually going to talk more about the paper. In this lecture, I'm going to give you kind of the background for this paper and a little bit of an idea of what they did, but mostly we're going to talk about the paper on Friday. And then also in this lecture, I want to emphasize a couple of questions that we're going to spend time in our class on Friday discussing. So let's actually start there at the end. If you look at the end of chapter 2 in the textbook, there are 11 questions listed for us, and I'm going to point out 3 that I think we're going to spend the bulk of our class time talking about from chapter 2, that is. First question is why could a double-stranded DNA break lead to structural chromosomal abnormalities? So that's question number 4. That's going to be one that you need to be prepared to be able to discuss on Friday. And the next one is, what is the explanation for the existence of mutational hotspots? And then finally, what is the connection between anticipation and nucleotide repeat mutations? So as you prepare for class on Friday, make sure that you know how to answer those questions, because we're going to spend time in discussion addressing those questions. Now, from the end of chapter 3, again, we have a list of questions. There are 10 of them. And again, I'm going to give you 3 that you really need to be able to address. One is explain the low incidence of monosomies. And then what are the different positions of chromosomal breakpoints? And then finally, what are the possible consequences of centric fusions? And so again, as you prepare for class on Friday, ensure that you know how to answer those questions. All right, now for the other point of this lecture, again, we'll cover a couple of topics we didn't quite get to on Wednesday, and we will get to some background on the paper that we're going to spend some class time on Friday discussing. All right, so some key terms. First, Mosaicism. This is one of the topics we didn't get to yesterday, and in Mosaicism, what you're looking at is collections of genetically distinct cells. And so within the same organism, you actually have different lineages of cells, and the different lineages are different genetically. Now, typically, we're not talking about huge genetic differences. We're talking about, small genetic differences between these cell lines. But some of the implications of this you can see in like huskies, where they've got one blue eye and one brown eye very commonly. That is a great example of genetic mosaicism. And we can see some other examples as well. Chimerism. So chimerism is, it's in some ways it's very similar to mosaicism in that you have genetically distinct cells, but in chimerism, the ancestry of those cells is different. They don't originate from the same individual. So you have within the same adult or child individual, you have cells that are from 2 or more different individuals, and we'll talk about how that happens in a slide in a little bit. This is Uniparental Dimerism, and so what happens here is basically your 2 copies of a particular chromosome, so let's say chromosome 1, are from the same parent, and so it leads to an unusually high level of unusually high level of, or let's say it this way, an unusually low level of genetic diversity in that chromosome. And we'll talk about how that might actually happen in a moment. All right, so inside of the chromosome in chromosomes that the centromere is not in the middle of the chromosome, you end up with a short arm and a long arm. The P arm, P stands for petite, is the short arm of the chromosome, and the Q arm is the long arm of the chromosome. Again, this notation works when you have chromosomes where the centromere is not in the middle and the two arms are not the same length, which is actually fairly typical for chromosomes. But this is helpful for this next term, chromosome mutation notations. And in here, we can represent chromosomal mutations fairly quickly and fairly easily. And so the most, or the ones that are going to come up in the paper that we're going to discuss on Friday are this notation here and this notation here. And so what this tells us is that we have a translocation of a piece of chromosome 4 over to chromosome 14. And so, you know, from our previous lecture where we had the Philadelphia chromosome, it would be noted this way, where you have a translocation of a piece of chromosome 9 over to 22. Now we get a reciprocal translocation, but the Philadelphia chromosome is this 9 going over to chromosome 22. This notation tells us that we have a deletion in the 17p arm on chromosomes, or on the p arm of chromosome 17. Could be the entire arm, or it could be most often just a portion of chromosome 17 that was deleted. All right, multiple myeloma. So again, this is focused on the paper that we're going to spend time tomorrow discussing. But multiple myeloma is a cancer of a specific type of white blood cell called plasma cells. And in multiple myeloma, these cancer cells accumulate. They begin to accumulate in bone marrow, and they displace healthy blood cells, and it disrupts the typical process going on in bone of wearing the matrix down and then remodeling it, and so it weakens bones and often leads to brittle and broken bones around the body. Another thing that you need to know about multiple myeloma is that multiple myeloma is what we call a heterogeneous cancer, and that we have different morphologies in the various cancer cells. And so that's where the multiple part of this name comes. And so, again, it's cancer in these plasma cells, and we get different morphologies in different cells. NDMM, this comes up a lot in the paper that we're going to discuss on Friday. It stands for newly diagnosed multiple myeloma. Now, another thing you need to know about multiple myeloma is it tends to be a cancer that is diagnosed very late in age. I think I forget what the exact percentages are, but I think it's about 90% of people diagnosed with multiple myeloma are older than 65. So doublet and triplet regimens, what these are, a doublet regimen is treating a cancer with two drugs at the same time. And then a triplet regimen is treating a cancer with 3 drugs at the same time. Now, what you find with multiple myeloma is that there are certain populations that make multiple myeloma particularly complicated to treat. And that's individuals that have this mutation or this mutation, and then there's a third mutation we'll talk about on Friday, that are particularly resistant to the typical doublet regimen used to treat multiple myeloma. And so those patients are given a triplet regimen and still have reduced survivability. And so the paper we're going to talk about, and I'll post on Canvas again, it's a review paper and basically goes through and looks at these different drug trials and treatment cases for patients with multiple myeloma and these specific cytogenetic abnormalities. All right. So let's talk about mosaicism in a little bit more detail. And again, in mosaicism, we've got cells, or at least a line of cells, a lineage of cells that are genetically distinct, that don't match the rest of the body. So here's the zygote. Here there's some kind of a mutation that happens in this cell, and then all of the cells that descend from that cell are now genetically distinct from the rest of the individuals. And here you now have this mosaic individual with genetically distinct cell lineages. And again, thinking about huskies with the brown eye and the blue eye as an example for this is a mutation that must happen prior to the eyes forming, and then that way the lineage that gives rise to the iris cells in one eye is different than the lineage that gives rise to the iris cells in the other eye. So mutations during the development will pass on to all the progeny of the mutated cells. So you end up getting this line that is genetically distinct from the rest of the organism. Now, we talk about mosaicism in our chapter in cytogenetic abnormalities or aberrations. And so if we're talking about chromosomal aberrations, we're talking about big mutations. And so we're talking about the potential to have, cell lines that are just completely different than the rest of the cells because of these large scale mutations. And a big one that can happen there is what we call mitotic nondisjunction. And so here, when you get nondisjunction and meiosis, it results in aneuploidy on some of our chromosomes in the gametes, either a gamete with two copies of a chromosome, which would then fuse with another gamete to give you a trisomy for that chromosome, or a gamete that's completely devoid of a copy of a chromosome, which would then fuse with another gamete and give you a monosomy. Either way, those are oftentimes devastating for the zygote. In mitotic nondisjunction, we basically accomplish the same thing in creating one cell. During a nondisjunction, one cell would get both, both sister chromatids for the paternal chromosome 1, let's say. And then so now one daughter cell from that mitotic division has only one copy of chromosome 1, and that would be the maternal copy. And then the other one will have three copies. We'll have the maternal and then two copies of the paternal chromosome 1. And so that's how you can get an aneuploid cell from a mitotic nondisjunction. And if you're wondering, well, can you have mosaicism in which some of the cells in your body have trisomy 21 Down syndrome and the rest of the cells don't? And the answer is absolutely, that can happen. Now, oftentimes these mitotic nondisjunction events happen later in development, but they can happen early enough in development to give that type of a scenario, although that is fairly rare. So that's mosaicism. Again, the existence of genetically distinct cell lines. Chimera Ism, again, the existence of genetically distinct cell lines, but in here, the ancestry of the two lines is different. And so probably the most common cause of this is when fraternal twins fuse in utero, and then so you end up getting cells from one twin it persisting in the twin that then becomes in some way the dominant twin, if you will, the only individual that actually survives. But some cells from their fraternal twin that fused with them in utero persist and become part of that organism and part of that individual, and so you get two genetically distinct cell lines present in that individual, but they came from completely separate people. Another thing that we've started to find is we first noted this because of the existence of cells with a Y chromosome in the uterus of mothers after they had had male children. And so it seems that cells from the infant will migrate out of, the developing infant and will sometimes migrate into the wall of the uterus, and then they will just continue to survive and sometimes divide and build these little pockets of cells that are lining the uterus that are genetically distinct from the mother, that they're actually some cells from her infant that continue to survive. We're also starting to find evidence that cells from the mother actually fuse with the infant while in utero and continue to be a part of that infant's body. Again, creating this chimerism where within the same individual you have two genetically distinct cell lines, and in this case, two genetically distinct cell lines that came from two different individuals. Last thing we're going to talk about is this. This is Uniparental Dimerism. And so, again, here you have an individual where both of their copies of a particular chromosome came from the same parent. And so this almost always happens when you get a trisomy in the zygote that is then fixed through various cell mechanisms. And so the cell has the ability to note that there is a trisomy in one of the chromosomes. Here in this example, we've got one of the maternal chromosome and then two of the paternal chromosome. And so one of the chromosomes is destroyed to account for that, to go back to a disomy, the condition that is normal. And here, if this chromosome gets destroyed, we basically get this situation. This is normal, one maternal chromosome and one paternal. If this chromosome gets destroyed, again, we get a normal cell with one maternal and one paternal. But if this chromosome gets destroyed, we end up with a uniparental disomy, where both chromosomes came from the same parent. And depending on when the non-disjunction event happened, if this happened for the non-disjunction event to create this gamete that has two chromosomes here, if it happened during anaphase 1, these two chromosomes inherited are likely to be separate chromosomes, because both homologous chromosomes get separated during anaphase 1, and if they end up being recruited together, there's a good chance that those chromosomes could still be unique, one from the paternal grandmother and one from the paternal grandfather. But if the non-disjunction event happened during anaphase 2, when the sister chromatids are supposed to be separated, these two chromosomes may be genetically identical. Both from the paternal grandmother or both from the paternal grandfather, in which case you would have an unusually low level of genetic diversity inside of these chromosomes, and it would make certain genetic illnesses that are inherited through some kind of a recessive inheritance pattern a lot more likely. And it just overall lowers the genetic diversity of the individual. So, again, as you prepare for class on Friday, make sure you know how to answer those 6 questions that I mentioned earlier in the video. We're going to spend a significant amount of time in class talking about that. We're also going to spend a significant amount of time in class talking about the paper and proposed treatment regimens for people with multiple myeloma that have some of these cytogenetic aberrations.